Good day everyone. Introduction to instrumental analysis. First, let us talk about chemical analysis. Chemical analysis is concerned in determining the identity of the substance or the amount of a particular substance in a sample. Now, when we talk about chemical analysis, it consists of determining or collecting sample, either treating it chemically or physically. After collecting, performing a laboratory or non-laboratory experiment, then manipulating the required data, and of course, get a meaningful result. Chemical analysis is divided into two. We have the classical and instrumental analysis. Now, in the classical or non-instrumental analysis, it requires the use of chemical, balance, calibrated glassware, and other laboratory apparatus such as beakers, burners, funnels, and flask. And one great example of the classical analysis, we have the precipitation, the distillation, and also the titration is also one of the classical method of analysis. Instrumental analysis requires the use of analytical instrument. So the analytical instrument uses or converts analytical signal into electrical signal. Then that is, electrical signal is being amplified into digital signal and then get displayed by the instrument. So ato ni siya tagsa-tagsaon aron masabtan gyud siya dayon. So this is how the analytical instrument works. So the sample, after the sample is being detected by the analytical instrument, so it uses energy stimulus, whether it is a current, light, or heat. So after which, analytical signal is being detected by the inducers or the detectors, then converted it into electrical signal. So the electrical signal is being processed or amplified, and then after to form a digital signal. So that digital signal is being read out by the instrument, then displayed in the form of digit. So that is how humans read or analyze the sample or data using the analytical instrument. Lastly, instrumental and classical method can be used in qualitative and quantitative analysis. Now, in the qualitative analysis, it yields about the information of atomic or the molecular species and the functional groups of the sample. Now, in the quantitative analysis, it provides information or provides numerical information that is relative amount or one or more components of the sample.
The first method of analysis is chromatography. Chromatography is the broad term for laboratory techniques designed for the separation or analysis of complex mixtures. The mixture is dissolved in a liquid called the mobile phase, which carries it through a structure holding another material called the stationary phase. The stationary phase remains fixed in a place while a mobile phase carries the components of the mixture to the medium being used. This stationary phase acts as a constraint on many of the components in the mixture, slowing them down to move slower than the mobile phase. The movement of the components in the mobile phase is controlled by the significance of their interactions with the mobile and/or stationary phases. Next method is capillary electroporses. It is a method of separating and identifying fragments of fluorescently labeled DNA by passing them through a very thin polymer-filled capillaries. The use of capillaries instead of a conventional slab gel makes it possible to apply a much stronger electric field, resulting in faster separation and higher throughout the process. It It is also used in science laboratories for the analysis of inorganic ions commonly encountered in post-blast residues. One is countercurrent distribution. It is a separation process that is founded on the principles of liquid-to-liquid extraction, where a chemical compound is distributed or partitioned between two immiscible liquid phases, oil and water for example, according to its relative solubility in the two phases. The simplest form of liquid-to-liquid extraction is the partitioning of a mixture of compounds between two immiscible liquid phases and a separatory funnel.
Cyclic voltammetry is a technique used to study reaction mechanisms that involve the transferring of electrons. The method involves linearly varying an electrode potential between two limits at a specific rate while monitoring the current that develops in an electrochemical cell. This image shows the cyclic voltammetry. Cyclic voltammetry is performed by cycling the potential of a working electrode and measuring the resulting current. So, the image represents the 3-electrode cell as used in cyclic voltammetry. A 3-electrode system is often used to separate the rule of referencing potential applied and balancing the current produced. So the next image shows the cyclic voltammogram. The result of cyclic voltammetry is a characteristic duck-shaped plot known as a cyclic voltammogram, which is seen in the slide. Mass spectrometry is an analytic method that employs ionization and mass analysis of compounds in order to determine the mass, formula, and structure of the compound being analyzed. Mass spectrometers can be used to identify unknown compounds via molecular weight determination, to quantify known compounds, and to determine structure and chemical properties of molecules. This image shows the mass spectrometry. So there are stages of what happens in a mass spectrometry. As atoms and molecules can be deflected by magnetic field, provided the atom or molecule is first turned into an ion, electrically charged particles are affected by a magnetic field, although electrically neutral ones aren't. So the stage 1 is ionization. The atom or molecule is ionized by knocking one or more electrons off to give a positive ion. This is true even for things which you would normally expect to form negative ions, such as chlorine, or never form ions at all. Such example is an argon. Most mass spectrometers works with positive ions. Stage 2 is the acceleration. The ions are accelerated so that they all have the same kinetic energy. The stage 3 is the deflection. The ions are then deflected by a magnetic field according to their masses. The lighter they are, the more they are deflected. The amount of deflection also depends on the number of positive charges on the ion. In other words, on how many electrons were knocked off in the first stage. The more the ion is charged, the more it gets deflected. The stage 4 is the detection. The beam of ions passing through the machine is detected electrically. This picture is the representation of mass spectrum. It is usually simplified into a stick diagram. In the analytical technique of mass spectrometry, atoms or molecules are ionized using a high-energy electron beam and then separated based on their mass-to-charge ratio. The results are presented as a mass spectrum, which shows the relative abundances of the ions on the y-axis and their mass-to-charge ratios on the x-axis. As you can see in the diagram, the vertical axis is labeled as relative abundance, while the vertical scale is related to the current received by the charge recorded, and so to the number of ions arriving at the detector. The greater the current, the more abundant the ion. Laser is an acronym for light amplification by stimulated emission of radiation. Laser is a type of light source which has the unique characteristics of directionality, brightness, and monochromaticity. So this image shows an example of lasers. A laser consists of three main components: a lasing medium, a resonant or optical cavity, and output coupler. The lasing medium consists of a group of atoms, molecules, or ions in solid, liquid, or gaseous form, which acts as an amplifier for light waves. For amplification, the medium has to achieve population inversion, which means in a state in which the number of atoms in the upper energy level is greater than the number of atoms in the lower energy level. The output coupler serves as energy source, which provides for obtaining such a state of population inversion between a pair of energy levels of the atomic system. When the active medium is placed inside an optical resonator, the system acts as an oscillator.
Microscopy is the technical field of using microscopes to view samples and objects that is invisible with the unaided eye, or objects that are not within the resolution range of the normal eye. In the field of microscopy, microscopes are the main instruments being utilized to observe micro-level living and non-living objects. Microscopy and analytical chemistry. Now, chemical microscopy, the combination of polarized light microscopy with microscale techniques of qualitative analytical chemistry, is an extremely powerful and versatile tool for solving a wide variety of contamination, particle identification, and material analysis problems. Therefore, in general, microscopy in chemistry is useful in observing changes of chemical composition or to determine contamination in the sample. Of course, extremely high-powered microscopes, especially the electron microscope, is one of the primary tools in observing these micro-level or microscale of particles. Now here are the sample images that can be seen under the electron microscopes. Now let us proceed with a spectrometer. By definition, the spectrometer or spectrophotometer is an analytical instrument used to identify or confirm the chemical species, chemical structure, or concentration of substance in a sample. The instrument emits an energy source to pass through a solution and measure light intensity at different wavelengths. If the solution is high in molecular concentration, then more light will be absorbed. Alright, now here in the diffraction and scattering techniques, we have the X-ray crystallography, the powder X-ray diffraction, and the X-ray diffraction. First, let us define the X-ray crystallography. X-ray crystallography is a scientific method used to determine the arrangement of atoms of a crystalline solid in three-dimensional space. This technique takes advantage of interatomic space of most crystalline solids by employing them as a diffraction gradient. Now here we have the powder X-ray diffraction is often easier and more convenient than single crystal diffraction, since does not require individual crystal be made. And lastly, the X-ray diffraction. X-ray diffraction, or the XRD, is a non-destructive technique that provides detailed information about the crystallographic structure, chemical composition, and physical properties of materials. So that's it.